my name is Soham, and my project is Decoding the Tumor Immune Landscape Across Cancers. Now, the role of the immune system in cancer isn't straightforward. Immune cells like cytotoxic T cells can identify and kill cancer cells, but at the same time, certain cells like macrophages can create an immunosuppressive microenvironment. Other tumor components like the extracellular matrix, layer proteins, and other molecules around the tumor can physically restrict immune cells, and signaling molecules can alter their behavior. The key idea is that these tumor immune interactions influence cancer progression and treatment outcomes, especially in immunotherapy, which uses your immune system to fight cancer. For immunotherapies to work properly, the patient's tumor must have the right conditions that enable the immune cells to actually target it. These factors collectively can be referred to as the patient's immune state. Now, some forms of immune state classifications exist. Immune hot tumors can have lots of cytotoxic T cells and usually have better outcomes, while immune desert tumors lack these T cells and have worse outcomes. The main problem is that we don't know what drives these immune states. We don't have a great idea on why a certain tumor has immune hot conditions while another is immune deserted. Understanding this is an important challenge as immunotherapy response rates remain low despite increasing over time. Nowadays, we have high throughput sequencing technologies like RNA sequencing, which allows a comprehensive profile of a patient's gene expression, which can be used for deep analysis of the tumor immune microenvironment. However, an issue is that many computational studies will focus on the impact of a single gene, which can overlook broader coordinated pathways. So what this project aims to do is to identify transcriptional programs, which are gene clusters, which we call metagenes, rather than single genes, that can represent the tumor immune landscape across a wide range of cancer types and patient samples. We will validate these clusters for their relevance to biological pathways and their prognostic value. Through this, we hope to shed some light on the mechanisms driving specific immune states and develop coordinated gene clusters for future biomarker development. To do this, we apply machine learning to bulk RNA sequencing data to identify these transcriptional programs and validate them with survival and gene level analysis. Now, onto the methods. We collect data from the Cancer Genome Atlas. From here, we collect bulk RNA sequencing data from a cohort of over 9,000 patients across over 30 different cancer types. We also collect clinical data available for these patient samples, like their overall survival, cancer type, age of diagnosis, etc. We also collect a dataset which maps each of the patient samples to an established immune subtype. These subtypes were developed by a team of researchers using this pan-cancer dataset. Now, to pre-process this data, we first impute missing values in the RNA sequencing dataset. Then we drop the bottom 40% of genes by variance, as genes with similar expression across all subtypes won't contribute much to distinguishing between different samples. Now, to select the actual genes that will form the clusters, we perform a one-way ANOVA test for each gene. So what this does is identifies the genes whose expression will differ the most across the different established subtypes. Why this step is important is that a gene whose expression is the same across all the subtypes wouldn't contribute to distinguishing between different immune states. We take roughly the top 5,000 genes by significance, and we use Gaussian mixture model to cluster the genes into 150 unique clusters of co-expressed genes. Now, these clusters are the metagenes, which I mentioned earlier. Our next step would be to use these metagenes to build a predictive model that classifies the established immune subtypes. This would see how well the metagenes could represent the tumor immune state. Now, not all of these metagenes would actually have relevance to the immune subtypes or biological pathways. We would have to select the best metagenes. These were the mean, median, and standard deviation of the expression values of each gene within the metagene. So from 150 metagenes, which are the gene clusters, we generated 450 metagene-derived features, which would be used to train the model. 3 models, XGBoost, support vector machine, to select the optimal feature subset from the 450 features. And Optuna was used to tune the model hyperparameters. The models were trained with 5-fold cross-validation, and training test metrics were obtained for each subtype. Now, the feature set from the best performing model was to be used for further interpretation. To analyze which of these metagene-derived features were the most influential on classifying these subtypes. We performed SHAP analysis. Now, what this does is it helps us identify how much each feature contributes to individual model predictions. We also go into a deeper analysis of these metagenes. We performed two types of survival analysis. First, Kaplan-Meier survival analysis was performed on the feature set used to train the best performing model. Essentially, what this does is it tests if the overexpression or underexpression of a metagene has any significant survival associations. Now, how this works is for each metagene-derived feature, the samples are split into high and low groups based on the median expression of the feature. So basically, the high expression group contains the samples that express the metagene greater than the median expression, and the low expression group contains the samples that express it less than the median. We then calculated the average 5-year survival probabilities between the two groups using the overall survival data from the clinical dataset, and adjusted p-values were calculated to test significance. On top of this, another type of survival analysis was performed. This can evaluate survival associations directly. So unlike Kaplan-Meier, you don't need to separate samples into two groups. So what's interesting about this is that it evaluates survival associations independent of any clinical factors. So using this, we evaluated the survival associations of the selected metagenes independently of clinical factors like gender, cancer type, and the age at diagnosis. Now, for the next two steps revealed the biological insights for the metagenes. For each metagene, we identified which genes were the most upregulated, and we also used a tool called G-Profiler to identify the biological pathways represented by the metagenes. Now, our results. So of the three machine learning models, XGBoost could best classify the subtypes with an 87% accuracy. Recursive feature elimination cross-validation selected 68 signature scores for this model, which encompassed 48 of the 150 unique metagenes. SHAP found that many of these metagenes were predictive of multiple different subtypes. Enrichment analysis with G-Profiler revealed that the top 12 metagenes, by the significance of the associations with existing pathways, mainly represented three themes: T-cell activation, cell cycle regulation, and extracellular matrix organization and remodeling. I'll go a lot deeper on the biological insights later in the discussion. Now, survival analysis showed many promising results. 40 out of the 48 metagenes showed statistically significant outcomes when overexpressed or underexpressed. Of these, 21 metagenes had an absolute survival difference between the high and low expression groups greater than or equal to 10%, and the top three metagenes had an 18% absolute survival difference. Cox regression provided promising results as well. 6 metagenes showed statistically significant survival associations independently of the patient's clinical factors. Now, this is important because it shows that the genes themselves have an impact on survival. Now, in the discussion, I'll go into depth on how the biological findings from the metagenes advance our understanding of immuno-oncology. Now, I can't talk about every single metagene, so I'll highlight 6 important ones. These 6 were chosen because of their predictive and or prognostic value. They together represent 3 key biological themes. So these two metagenes 147 and 82, represented a framework of coordinated immune regulation in tumors. Metagene 147 represented a balance of immune activation and suppression in tumors. On one hand, it had a lot of MHC2 molecules, so then its overexpression would appear to allow immune cells to better identify cancer cells. However, at the same time, co-expressed with these molecules are immunosuppressive genes. For example, PDL2 is an immune checkpoint molecule. Now, metagene 82 was quite interesting. G-Profiler enriched it for a defense response to a virus. Upon further analysis, it contained a lot of interferon-stimulated genes, which are expressed in both virus-infected and cancer cells. Though these molecules typically combat infection, if they're chronically expressed, they can actually promote tumor progression. So the key takeaway from these two metagenes is that even if a tumor appears to have strong immune activation, they may also co-express suppressive mechanisms to evade the immune system. This can explain why using just single gene biomarkers like immune checkpoint expression or T-cell counts may fail. So effective immunotherapy also needs to block these suppressive mechanisms. Now, the main idea of this project was to elucidate immune states in tumors. So at first, it was a bit odd that cell cycle regulation played such a big part in this. On the surface level, it doesn't seem to be related that much to tumor immunology. But analyzing these two metagenes revealed that cell cycle regulation in tumors has immune consequences. Both metagenes had genes regulating mitotic checkpoints, which from the literature had associations with immune infiltration. They also both had genes for DNA replication and repair, which can both directly and indirectly influence tumor immune populations. Now, the key takeaways from this are that immune infiltration and tumor proliferation shouldn't be seen as separate aspects of cancer. They're dependent on each other. This supports existing strategies which target cell cycle machinery to improve immunotherapy outcomes. Now, some of the metagenes with the strongest impact on survival outcomes were these two, related to the extracellular matrix and the surrounding components of the tumor. Metagene 78's main finding was that the breakdown and strengthening of the extracellular matrix are co-expressed in tumors. This metagene had a lot of collagen-coding genes, which will thicken the extracellular matrix, which would physically block immune cells from infiltrating. But at the same time, it had a matrix metalloproteinase, which will break down the extracellular matrix to allow cancer cells to invade other tissues. Now, metagene 75 was quite interesting. Its overexpression was significantly associated with worse survival from both survival analysis techniques. Enrichment analysis with G-profiler didn't show any strong direct associations with microenvironment pathways. It was actually enriched for the negative regulation of apoptosis, which is programmed cell death. But from analyzing the top upregulated genes, we saw how this biological program combines multiple pathways for effective immune evasion. First, like metagene 78, it simultaneously expressed extracellular matrix cross-linking and degradation. It also included a lactate transporter, which would export lactate from cancer cells and make its surroundings highly acidic. This would impair the proper function of immune cells. On top of that, it also contained some molecules that directly suppress the immune system by influencing the numbers and activities of certain cells. Basically, this combination of physical, metabolic remodeling, and direct immune suppression is likely to be a fundamental mechanism of tumor progression. So now I'll cover what's so significant about these metagenes. Well, first, these 48 unique metagenes can classify immune subtypes across many different cancers and samples with high accuracy. This shows that they can represent the different aspects of the tumor immune microenvironment and distinguish between patient samples. More than just being predictive and explanatory, they are prognostic as well. The expression of many of these metagenes influence survival outcomes even after adjusting for clinical confounders. As a result, they may be the foundation of future biomarkers, as they're more robust than just single gene biomarkers and are generalizable across many cancer types. The biological insights, which I talked about earlier, may help uncover novel regulatory mechanisms of the tumor immune microenvironment. So for the next steps, the main priority is to validate the survival associations on independent RNA sequencing datasets. Furthermore, this project evaluated the metagenes across over 30 different cancer types, but targeted analysis of the metagenes specific cancer types should be performed to understand how their impact will differ across certain cancer types. Another thing is that this project analyzed the metagenes with bulk RNA sequencing data, but single-cell RNA sequencing data could provide a deeper understanding into what cell types express the genes of the metagenes and how it would affect survival outcomes. So now just a final recap of what we did and what we achieved. Unsupervised clustering of pan-cancer RNA sequencing data yielded 48 immune-related metagenes. Supervised classification and SHAP show that the metagenes could accurately represent the tumor immune microenvironment. Survival analysis revealed that the expression of many metagenes had significant survival associations, and lastly, gene level and enrichment analysis highlighted their biological pathways, advancing our understanding of immuno-oncology. Like I would say like my mentor, Vamika, she was like really helpful in like guiding me through like the methodology and especially in like the actual writing process. Like her experience really helped. So like this helped me developing a research plan and like presenting it and like writing in a paper, which is something I hadn't done before. I hadn't had much experience, but like YRI helped like structure like the writing process and research process, which like really helped me.